Hey students, so today I am showing you how to transcribe and translate a DNA code so that you know what amino acids are being produced and what protein gets produced, okay? So this is going to be very similar to what you're going to see on the test. So what you'll do first is you'll label your DNA template strand. So this is our template strand right here, and I'm just going to write temp on there, okay? Just to help me remember that that's the one that helps determine what the DNA complementary strand is. So I'm going to put comp right there for complementary. And then it also helps determine what my mRNA strand is, okay? So I like to draw an arrow from my template strand down to my complementary strand, and then also an arrow down to my mRNA strand, just to help me remember that this template strand is what determines both the complementary strand and the mRNA strand. Okay, so remember that the DNA has to stay in the nucleus. We don't want it to get damaged, so we're going to keep it safe in the nucleus, but we still have to get this code out to the ribosomes to be able to make proteins. So the first thing we do is we're going to just write out our complementary code, and that's gonna just be regular DNA complements, okay? So across from every T we'll have an A, and across from every A we'll have a T, across from every C is a G, and across from every G is a C. Okay, so that would be my complementary code. And then the next thing we're going to do is we're going to make sure we know what our mRNA code is, okay? So across from a T, if I open up this DNA strand and use my template to make my mRNA, across from the T I'll have to have an A bound to it. But the difference between DNA and RNA is that across from the A's, I have to bind a uracil. So remember that uracil is used in mRNA instead of thymines, okay? So the mRNA code is going to be very similar to the complementary code, other than where there's a T in the complementary code, I would have a U in the mRNA code. So we're just going to finish filling that in. And right here, I've got a T there, so that means across from the adenine, instead of a T in the DNA, I would have a U in the RNA, okay? Same here, U goes there. Okay, so that's my mRNA code. And then okay, so I've got my mRNA strand made. And the next thing that we're going to do, and this doesn't really affect a whole lot of what you're going to be doing as far as the amino acid, but the next thing we want to do is just figure out what our anticodons are going to be on the tRNAs. So I also like to draw an arrow from the mRNA strand down to the tRNA strand, and another arrow down to the amino acid strand, okay? So mRNA is what determines both the tRNAs and the amino acids, okay? So across from the codon AUG, I would have UAC, okay? And I like to take, especially in mRNA, I like to think of them 3 at a time. So the next codon would be CGC, the next codon, or the next anticodon in the tRNA, across from the AGG, which is the codon. This would be a UCC, and then a GCC, and then a GAG, and then an ACU, okay? Now, to figure out my amino acid, I need to make sure that I'm looking at the codons. So remember that codons are on the mRNA strand, and I'm just going to put a little box around this codon right here, because that's going to be the one that I want to use first. So we take that codon and we look at our decoder sheet here, okay? And our first codon was AG or AUG. So we always take the first letter that's the biggest here in our decoder, and we go work our way out, okay? So AUG as a codon would give me methionine for the amino acid. So back here on my sheet, I'm going to write down methionine. And there's not a whole lot of room to write out the whole name for methionine, so I'm just going to abbreviate that to METH. You could, a lot of textbooks will have you just put MET in there, okay? So then the next codon is GCG. So I'm going to look at my decoder sheet here and work from inside to outside. So G, C, and G. And you'll notice that there's 4 different codons that can code for that alanine right there. But we're looking specifically at GCG. So alanine is my next amino acid. And I'm just going to write ALA for alanine. Okay. And then my next codon is AGG. And AGG codes for arginine. So ARG for arginine. My next codon is CGG. C, G, and G, so another arginine. And you can have lots of the same type. There's only 20 amino acids, so you can have lots of the same combinations in different proteins. But so what determines the protein is the order of the amino acids, and then also the way that the protein is folded. So I could have the same order of amino acids, but it could get folded differently and it would be a different protein, okay? Sorry, quick side note there. Okay, my next codon is CUC, which is gonna be CUC, so leucine. And I'll just write my LEU for leucine. And then my last codon is AGA, and this one's usually a special codon that basically tells the ribosome to stop making the protein. UGA. So we're going to find UGA, and you'll see that that says stop on that one. So we don't actually have an amino acid here, so I'm just going to put an X through that and write a stop next to it, because that's our stop codon. That's the codon that says this amino acid's done. So I hope this helps you. Go through the next problem. On this next one, you'll have to work backwards from mRNA. I think it's best to take that mRNA strand and really figure out the template first, and then you can figure out the complementary DNA strand, and then the tRNA strand, and then the amino acids. So I hope this helps. And good luck with your transcription, translation, and protein synthesis.